Good morning or good afternoon, depending on where in the world you are today. Welcome to our webinar, From Patents to Treatment, Using IP Intelligence to Retarget Molecules in the Fight Against COVID-19. I'm Emma Barry, COO of Ascensio, and I'm delighted to welcome you to this, the second in our series of webinars, where we discuss our COVID-19 initiative and the use of our technology against this terrible disease. Since the pandemic began, the team here at Ascensio has been working to do whatever we can to help the worldwide effort to find effective treatments. As our IP-based molecular landscaping approach is so different, we realised that there was a chance at least that we may be able to identify treatments that other medicinal chemistry approaches may not find. Our session this morning is focused on this retargeting work. We will be discussing our approach and our results so far. Our speakers will be Kevin Brown, our President and CEO, and Kevin Brogle, our Chief Scientific Officer. So, without further ado, I'd like to hand over now to our first speaker today, Kevin Brown. Kevin, over to you.
Good morning, and thanks for joining. My name is Kevin Brown. I am the President and CEO of Ascensio. Special thanks to our partners. You can see them there at the bottom of the page. So, in terms of the overall initiative, what is it and what are we doing? So we're using our technology to do two specific things. On the left-hand side is retargeting. So that's basically identifying clinical stage and marketed molecules that can be retargeted to the COVID effort. These are molecules not necessarily indicated or related to COVID-19 biological targets, but through the use of our technology can be. And on the right-hand side, we are providing our technology to medicinal chemists and other researchers so that they can use it to generate new molecules that may have COVID-19 potential. As Emma stated at the beginning before she fell out, the focus of this discussion is the retargeting effort. So we'll jump into that. In terms of retargeting, there are three basic outcomes. Of course, if we identify a molecule or a set of molecules that can be retargeted or that show some level of promise using our tech, there's immediate potential for those molecules. But there are two other outgrowths from this particular activity. One is around new molecule discovery. So now these retargeted molecules become seed scaffolds where chemists can now take them and do their chemistry work, create chemistry action plans around these new scaffolds that have been retargeted into COVID-19 related biological targets. And the third piece, which is also really interesting, is the original targets where these retargeted candidates came from now begin to show, or potentially there is a window between that original target and the COVID-19 related biological targets, and maybe there are other molecules that can transition over and back and forth and so on and so forth. So what have we found so far? Ascensio has identified 120 potential antiviral molecules and 26 potential host target molecules. And I say so far because this is an ongoing effort. You'll see that as we go through this presentation, that this is a continuous process. Our current target roles include the initial immune response, viral replication, viral invasion, and the cytokine storm. And we've developed a prioritized list of these hits. We'll be doing some biological testing to confirm some of these hits. Now, there are other organizations doing retargeting or repurposing, and this is, of course, an ongoing effort across the globe. But what makes Ascensio's effort unique is this. So we're using this tool, which is called IP-GeoScape, to find these retargeting candidates. And I'll talk to you a bit more about this in just a second. But for context purposes, who is Ascensio? So we're a tech company driving innovation through the use of intellectual property. IP is the medium through which we view all questions and seek to answer all questions. And we're AI and machine learning based. But the guiding principle behind our company is that no idea should be wasted. And what do I mean by that? So seeing and harnessing the full potential of intellectual property is difficult. Here's an interesting stat. Within the biopharmaceutical industry, 90 to 95% of all industry patents and applications never produce value in the form of marketable products or other related revenue. Now think about how much waste that is, and that's just for the ideas that the industry thinks is worth it to put into a patent or patent application. So at Ascensio, what we've done is we've turned this on its head. We use intellectual property as the lens or as the medium through which we view other information. So we look at curated data and compiled intelligence. We develop actionable insights and specific next steps, determine monetize IP opportunities, but all through an intellectual property lens. So what do I mean by a lens? We call it intelligence through an IP lens. Wherever there's a question that needs to be answered, whether it's an R&D based question or a legal question or a business development question, if we're talking about a biological target or an ion channel, a therapeutic area, it really doesn't matter. What we do is we create a set of visualizations or a visualization that illustrates the similarity between the IP in question. So the IP that we're going to use to try to answer that question, we create a visualization that illustrates the similarities that exist between that IP. The second piece is that we add data to the visualization. So all of our visualizations are data agnostic, meaning that any data can be added to it, whether that's private data from a customer or publicly available data or data from a vendor, it doesn't matter. If we're looking to answer an R&D question, maybe that data is biological activity. If we're looking to answer a business development question, maybe that's the financial information associated with a patent owner, that type of thing. And at the end of the day, we take those visualizations with the added data, and either we use them or our customers use them to answer their business questions. So one of those tools that I mentioned previously is IPgeoscape. So the IPgeoscape lens is a visual landscape of molecular IP space. It shows the structural relationships that exist between molecules, and we do this for both small and large molecules. And it's very easy to interpret. It looks like it's a lot going on here, but let me explain it to you. So what you're looking at here is the IPgeoscape for the biological target IRAK4. How do we create this? We identify all the players in the area. So all the companies, all the universities, any entity with a patent or with patent applications claiming activity against IRAK4. Once we identify all those organizations and all those small molecule patents and patent applications, we extract the small molecule information. And specifically, we extract the exemplified molecules from those patents. We stick it into our tool, and this is what pops out. Each color here represents a specific assignee. So a specific university, company, doesn't matter, anyone with patents in this area. The colors represent those different entities. So there are 23 different entities in this landscape right now, 23 different colors. Each marking represents one specific molecule associated with that particular assignee or entity. On this map right now, there are over 11,000 exemplified molecules. The shape and the color represent a specific molecule being disclosed into a specific patent or patent application. So I've circled this blue square here. So that would represent that molecule, number one, belonging to Gilead, blue, being shaped by the patent family, which is 2368, and being associated or being this particular structure right here. And the map is very easy to interpret. The only thing that you need to remember is that molecules are clustered together based on structural similarity. So the closer molecules are to one another on this map, the closer they are structurally. The further apart they are, the more different they are structurally. So in terms of COVID-19 and our focus, this table shows the initial targets currently being analyzed by Ascensio. So we've done all of our retargeting analysis on the targets that you see here, and you can see the various target roles. And as I mentioned to you previously, we made some of these landscapes available to researchers, and they're using them right now to guide some of their medicinal chemistry efforts. And that's here through our friends at Perkin Elmer, who've partnered with us on this effort. This is the Perkin Elmer COVID-19 Analytics Resource Center page. And if you were to go here, this is what you would see. And you can see the data that's associated here, various featured content, Ascensio being one of them. And so one of our targets here, I'll show you, for example, RNA polymerase inhibitors and what that looks like. When you click on that, this is what you'll see. Of course, there's a landing page, and you can see kind of what's going on there. And of course, some information in terms of using the landscape. But this here is the RNA polymerase inhibitor landscape. So you're looking at all the companies, all the universities, anyone with patents and patent applications claiming RNA polymerase inhibition. And they're colored by the various companies. So for example, this orange color is shearing, this blue color over here is Japan Tobacco, and all of these things are active. So each individual marker is a molecule that you can pull up. And of course, molecules being close to one another are more similar. It makes sense that Japan Tobacco or shearing right here, all their molecules are clustered together because they're very close. And then, of course, molecules that are much further apart are very different structurally. That's how you interpret the map. This is the lens. And I mentioned that there's additional data that we add. That's over here to the right. So you can see, for example, all the companies. Maybe you want to see the AbbVie footprint, or maybe you want to see another company's footprint, or maybe you want to see BI with it interdispersed. So these are all things that medicinal chemists, they're using it right now to guide some of their research efforts, information about the patents. Maybe you want to understand how the entire area developed over time, starting in 1994, as the landscape grew, as other companies began to enter the area, seeing various trends, seeing certain hotspots, things of that nature. But the obvious question is, well, Kevin, I see here that you have a molecular landscape for RNA polymerase inhibitors, and I understand it's showing the structural relationships that exist between those molecules. But how do you use this to identify clinical and marketed molecules not claiming inhibition against RNA polymerase, but that you're saying your tool helps you find that potential? So to answer that question and to give you more details, I'm going to turn it over to my partner, Kevin Brogel, who is Ascensio's Chief Scientific Officer. He's going to walk you through the details of that, how we came to look at the various targets, and even go through some of the molecules that we found. So with that, Kevin, take it over.
Thank you, Kevin, and thank you all for joining me. I'd like to start by talking to you about our process, the why and the how. Two of the most common approaches to retargeting have been in vitro screening and structure-based predictions. While these approaches to retargeting are valid and important, they, as well as ours, have different benefits and risks. Because of this, Ascensio believes that the retargeting of clinical molecules, particularly when addressing a pandemic, should involve the use of multiple orthogonal approaches to ensure a thorough retargeting attempt has been made, and that our approach is an important part of that. Now, onto the process itself. The first step was target selection. Targets were selected based on two criteria. First, we wanted to cover a variety of ways to affect the disease. And you can see in the table on the right, the different roles. Brown had mentioned these earlier: viral replication, viral invasion, the initial immune response, and of course, the deadly cytokine storm. The second criteria is that there also had to be sufficient patented molecules to produce an IP-GeoScape. Now, some of the targets had too few patented molecules for use in medicinal chemistry, but had enough to be used for retargeting efforts. In the case of the viral targets, I do want to mention that no molecule specifically designed for SARS-CoV-2 had published patents. So we prepared universal RNA-dependent RNA polymerase and viral protease IP-GeoScapes. While this is not our usual approach, it is the best option available, and the homology between the proteins may allow for some cross-reactivity and perhaps may lead to broader spectrum inhibitors. The next step involves the clinical molecules. We began with a database of more than 14,000 molecules that at a minimum reached clinical testing. We reduced these to a set of more than 6,000 unique structures for retargeting. The use of our IPgeoscape along with our scoring regimen allowed us to minimize the risk of what's called the curse of dimensionality. This is a phenomenon inherent to high dimensional data analyses such as molecular similarity. The final step, as Kevin Brown said earlier, will be the confirmation of our retargeting efforts via in vitro pharmacological testing. We intend to produce full concentration response curves in order to avoid the false negatives associated with high throughput screening. This table shows the current status of the retargeting efforts for each selected target. 7 of the targets resulted in virtual hits, with only IL-1 and TLR3 resulting in no hits that met our criteria. Some of the hits are already known to interact with that target. That's the second row here. Though others are brand new, that's the bottom row. These are molecules that are not previously associated with the biological target of interest. While we await confirmation via the pharmacological testing, we do have some evidence that our process is effective. 3 of the 60 new RNA polymerase hits have been confirmed via a separate independent retargeting effort involving screening of the reframe library. Now, while we do not yet have biological data to share with you, I would like to share with you a few select examples of the new hits that are awaiting confirmation. The full list will be available on our website, and Emma will show you how to access that later. Here you can see the basic information about a few of these. On the left is the retargeting target. That is the biological target that's involved with COVID-19 that we want to retarget our molecules to. Then we have the drug name, the structure, the original target, if it was known, of some of these retargeted molecules, and their highest clinical status. And we have some examples here from ACE2 to TLR7 to IL6. And you can see on the right, they come from a variety of different original targets and had reached the different stages of development, ranging from phase 1 clinical testing all the way to marketed molecules. With that, I'd like to give you a short demonstration of our IPgeoscape technology and how we're using it to retarget these clinical molecules. So here you can see the TLR7 agonist IPgeoscape landscape tab. It's very similar to the one Kevin Brown showed you earlier regarding RNA polymerase. It has fewer molecules in this case, but the concept's in the same. They're colored by the assignee. Each market is a different exemplified compound extracted from TLR7 agonist IP publications. The examples, of course, as he said, are clustered together based upon similarity, and that's using our proprietary molecular numericalization scheme, and the clusters are grouped on a similar basis. You'll also note that there's unoccupied space represented by these empty clusters. I'll speak a little more about those later. When you see markers of different colors in the same cluster, it means that more than one assignee are working on similar compounds. And you can see that here with these two molecules. Even if you're not a chemist, you can see how similar these two molecular structures are to each other and why, even though they're from different companies, they're being grouped together. You can also notice the landscape is completely interactive. There's the filters on the right that Brown showed you earlier. And I do want to add that the IPgeoscape is data agnostic. So any data connected to the assignee, the patent family, or the compound can be incorporated and used to filter. Now, while the landscape tab is our primary display, we do have other that provide different views and capabilities. The competitive overview tab shows the who, the what, and the when, and provides competitive intelligence. In the upper left, you can see basic information about the GeoScape, the number of patent families, the number of assignees, and the number of molecules it contains. At the top is a bar graph showing each assignee and the number of patent families they have. For instance, AstraZeneca has the most with 15, and Roche is a close second with 14. On the left is another bar graph showing the breakdown by priority year. This shows you how interest in the target has changed over time. In this case, interest has been steadily increasing since the first priority year of 2003. In the middle is the intersection of these two bar graphs. You can see when each assignee was active in any particular in this particular area. For instance, AstraZeneca, while they have the most patent families, actually hasn't been active since 2012. These charts are all interactive and can be used to filter the patent table at the bottom. I do want to inform you that the patent family number in the table is hyperlinked to Google Patents, and this enables you to review the claims, the specifications, and the status of the patents as needed. The Analysis tab lets you look at overlaps in the intellectual property. Let's clear this for you. Here, each cluster is reduced to a stoplight colored tile. You must select a particular assignee, such as AstraZeneca, in order to view the tiles related to them. Green tiles indicate exclusive space. You see when I select this green tile, over in the table on the right, only AstraZeneca patents are shown to have compounds in that particular cluster. Red tiles indicate overlapping space. So here, when I select this one, you see that compounds from 5 different patent families from AstraZeneca are present in this cluster, but so are compounds from 2 other assignees. Then finally, yellow tiles indicate near neighbors. Here, the selected assignee, AstraZeneca, has no compounds in this cluster, but molecules in that cluster are very similar to the neighboring molecules from our selected assignee, AstraZeneca. So these selected clusters, these compounds from Janssen will be very similar to these neighboring molecules from AstraZeneca. We also have our Medicinal Chemistry tab. This provides another view, one useful for molecular design. Here the view is again tiled. Each tile, when selected, displays an image of the most representative structure from that cluster, which has been highlighted by the most common substructure. This gives a simple but powerful representation of the contents of that cluster. Selecting a tile also results in the table on the right, filtering to all the compounds that are in that cluster, providing also all the associated data with the molecules in that cluster. I'm not going to talk in depth about how medicinal chemists use this tab, but I do want to point out that you can clearly see that unoccupied space I mentioned earlier. These are spaces that are ripe for new molecules. Now back to the retargeting. I'm going to select this clinical clusters tab, and you can see here again the same molecules with the same colors as were on the landscape tab. We've simply changed the shapes to all circles in order to make them contrast with the overlain clinical molecules. I'm going to overlay those now, and they're showing up as these white X's. All 6,000 plus are added regardless of fit, but the fit score can be used to filter those with the highest likelihood of successful retargeting. I'll briefly remove the patented molecules so you can see at this particular fit level, there are 21 remaining molecules, clinical retargeted molecules. Now, most of these are known TLR7 ligands, such as metolamod down here in the lower left. Metolamod is a known TLR7, TLR8 agonist. It's from Array Biopharma, and it's clustered here with other patented TLR7 and 8 ligands from Array Biopharma. You can see the close similarity of the structures because they're from the same patents. However, some of the molecules are not previously known to interact with TLR7. One such example is BIIB021, which is this molecule here. It's a heat shock protein 90 inhibitor, and it's clustered together with similar structures that are TLR7 agonists from IP from Janssen. Another retargeted example is tenofovir. Tenofovir is a reverse transcriptase inhibitor, and it's clustered together with some similar molecules that are patented TLR7 ligands from Gilead. Now we've done this for we've made these IPgeoscapes available for 5 different targets. I'm not going to take you into all 5 of them, but I do want to show you one more, not just the TLR7, and that is CXCR3. Here again, you see the landscape tab. It's interpreted the exact same way as the RNA polymerase and the TLR7 agonist ones we've shown you earlier. So I'll just go straight to the clinical clusters tab. And I'm going to overlay those 6,000 plus clinical molecules again. You can see they fall in different spaces, of course, than they do on the TLR7 agonist IP-GeoScape. And we're going to filter those down to the ones, again, most likely to have be successfully retargeted. In this case, there's a handful of them left. Again, most are known ligands. This should be unsurprising. For example, down here, we have PS386113. This is a known CXCR3 ligand from Pharmacopoeia, and it's clustered together with the rest of Pharmacopoeia's CXCR3 patented molecules. Another interesting one is Borixafor. Borixafor is a CXCR4 antagonist that is clustered together with other patented CXCR3 and 4 ligands from Tygen. So I think this helps you understand a little about our IP-GeoScapes, how we're using them, and how we're using them to retarget molecules. I'm now going to hand you back over to Emma Barry, who will continue the presentation. Emma?
Thanks very much, Kevin. Hope everybody can hear me now. So, in summary, our retargeting work so far has identified a total of 120 antiviral molecules and 26 host target molecules, which have the potential to become immediate candidates as COVID-19 treatments. What we've done then is we've developed a prioritized list of these hits for confirmatory testing via biological screening, which of course is the next important step in this initiative. Once that screening data is available, we will of course include that within the COVID-19 IP-GeoScape Library, which forms part of the Perkin Elmer Analytics Resource Centre, which Kevin Brown showed you earlier. But really this initiative is only just getting started, and we will be adding to the library and our retargeting results. So we do really encourage everyone to keep in touch with us and also check out our website, which we will be updating on a regular basis as and when we have new information to share. So I just want to quickly show everybody how best to access that and indeed access the full list of retargeting results. So I'll just take everybody to the website now. So this is our homepage, ascensio.com. And then to access anything associated with our COVID-19 initiative, you just need to click up here to access our COVID-19 page. From here, you can catch up on any of our previous webinars, and as we introduce new ones, register for the new future webinars as well. As I mentioned, you can also access the Perkin Elmer Analytics Resource Centre by clicking here in the new molecular research section. But the thing which is most interest to this session today is here. This is how you access the retargeting results. You just click here and that takes you to this page. So this page lists all of the results for all of the available targets right now. To access the data for each individual biological target, all you need to do is just click the target name. And as you'll see, that brings up a table, which is scrollable and shows the full, for example, here, 18 hits that we've discovered for ACE2. So if remember, Brogle mentioned Requinimex, and there it is. To then collapse, you just need to click again. And then as you can see, as we go through, each of the structures are shown. So if I just click TLR7 agonists here, and I scroll down, you'll see tenofovir, which is the already launched chronic hepatitis B and HIV treatment, which we've identified as possibly being of interest for COVID-19. So what's really important to us is that this is a community effort. It really is a collaborative effort. So we are really encouraging of anybody who's got suggestions, ideas for new targets, to get in touch with us. And also, if you are working on biological screening yourself and you screen any of the molecules that we've identified, we would love you to share that information with us. And you can do that here on our page just now. But on the subject of sharing and being collaborative. I believe I saw a number of questions. So what I will do now is I will just click over to the Q&A section and I will start to go through those. So I think I saw this come in quite early when we were first demoing the geoscapes, but we have the question, do the X and Y axes represent anything in the landscape?
Emma, I'll take that question. No, they don't represent anything in particular. They are arbitrary axes. They are just used to arrange the clusters in relation to each other.
Great, thanks. Now, our next question is, are you looking at antibodies as well as small molecules in your COVID-19 analyses? You say IPgeoscape technology can be used for small and large molecules. Are there large molecules being evaluated for COVID-19?
Yeah, while our IPgeoscape technology can and certainly is used to visualize large molecule IP landscapes, antibodies are too specific for retargeting. So we have not done any antibody-based IPgeoscapes as part of our COVID-19 initiative. There are, however, fortunately, many antibodies in clinical trials to treat COVID-19, some of them targeted towards the very targets that we are including, such as IL-6.
Thanks very much, Brogel. Before I go on to the next couple of questions that have already come in, I would encourage everyone, please do, if you've got any questions at all, don't be shy. You can enter your questions down in the Q&A section, which should be at the bottom of your screen. I'll move on to the next question, which is, are you considering any other biological targets in addition to the ones you listed?
Yes, we are. We're constantly reviewing the literature for potential additional targets. And as Emma suggested, we are soliciting ideas from the vast COVID-19 world community as well. And they will be released whenever they come available.
Great, so we've got another question here. Why is your IP-based approach so powerful? And is it different to other approaches to retargeting?
I think the main point of it is that it's different. When you're looking to retarget molecules, you want to be as thorough as possible. The one false negative that you get may be the one molecule that can be retargeted. So we think it's best to take different approaches using different techniques. That way you're going to ensure that you've taken the most thorough effort and you've not missed a key retargeting opportunity.
Great. We'll move to the next question, which is, how does retargeting differ from drug repurposing? And can you use the same tool for repurposing?
Drug retargeting is kind of a subsection of drug repurposing. Drug repurposing is taking a molecule that's made it at least to the clinic and changing its indication to something it wasn't originally intended for. That can involve keeping the mechanism occurring through the same biological target or retargeting it to a different mechanism, a different biological target. We tend to use retargeting because we're not just repurposing. We're not changing the indication, but keeping the target the same. We're changing the biological target and potentially opening the window to a new set of indications for that molecule.
Great. And then we've got another one, which is, what's the status of the other viral targets listed, such as RNA polymerase?
Yeah, you may have noticed those aren't up on the website yet. We are currently finalizing the retargeting results there, and those should be released and put up on the website shortly.
Great. And I think we might have one that's just come through on chat as well. How often do you update the patents, the data?
We update them quarterly.
Great. Now, I will, as they've been coming through on chat and Q&A, I will give everybody just a little bit more time. So I would encourage anyone, if you've got a burning question, feel free to type it. whilst we're waiting for that, I'll do a little bit of the thing that unfortunately I couldn't do earlier on, just to let everybody know. As you've probably seen from the little button up in the corner, this session is being recorded, and you will be able to access the recording either via our website, as I mentioned earlier, or we'll also be sending out a link in a follow-up email after this. Just going to see if there's any more questions. Nope, so it looks like we've covered all of the questions raised here. But again, oh, hang on, no, we've just had another one come through. Oh, how do you extract the patent structures?
We work with one of our partners, actually shown here, Chemaxon, to extract the structures from the patents.
And I was a bit quick before and almost missed that question. So if anybody has any other questions. What is the cost to your program?
Brown, why don't you take this one?
I'll take that. I was actually on mute. So we offer the IPgeoscapes, whether it's the COVID-19 IPgeoscapes that are available. That's complimentary through our friends at Perkin Elmer. The IPgeoscape in general is offered as a library, and you can access them for about $5,000 a piece, and there is a seat license, and you can just buy them at your leisure. And we do offer them in a customizable fashion as well. And that's on a case-by-case basis. Maybe there's a biological target that you're interested in and you want to have an IPgeoscape created, or maybe there is additional data that you'd like to see added, maybe some of your own molecules that you're creating in-house, that type of thing. So that's the difference between the two. And that's on a case-by-case basis, and the cost will vary on a case-by-case basis.
Thanks very much, Kevin. So if anybody does have any more questions, if they don't pop up in the next minute or so, please do feel free to follow up with us. You can do so either through the website or directly in that follow-up email I mentioned a minute ago. We will be putting our contact information there. So, we really love to hear from anybody if you've got any additional questions or you want to dive into anything that we've discussed today in a bit more detail. If there's no other questions, all that remains to be said is thank you very much to each of our initiative partners, which you'll see their logos listed here. It's Perkin Elmer, Clarivate, Chemaxon, Invensis, and Ben Franklin Technology Partners, Southeastern Pennsylvania, without whom this initiative couldn't have become a reality. And of course, a big thank you to you all for attending. Have a lovely rest of your day, and goodbye.